We're interested in developing new strategies to cure and prevent HCV infection in order to overcome some of the limitations associated with existing therapies. In this aim, we've developed a monoclonal antibody that targets one of the host proteins involved in HCV infection. And so this antibody is able to both block infection of cells, and it also was able to cure chronic infection in monotherapy in mouse models for HCV. We know that not all patient groups can be reached by current therapies, particularly people with genotype 3 HCV, and also in the context of graft reinfection following liver transplant and chronic liver disease caused by chronic HCV infection. The current therapies don't always work well for this. And so our goal with our antibody is to be able to reach these patient groups who currently don't have good therapies available. We first produced this antibody in rats, and we then characterized this antibody in cell culture models, primary liver cultures from patients, as well as the mouse models that are available for HCV. The antibody showed really great activity, but we can't use a rat antibody in humans, so we had to humanize the antibody, which was actually the study I'll present at EASL this year. So we essentially took the relevant sequences from the rat antibody and pasted it onto a human backbone, antibody from a human antibody backbone. And so we've then been characterizing this humanized version of the antibody, and it also has the same activity. It prevents infection of all HCV genotypes, and it can also cure HCV infection in mouse models. The surprising thing was in our mouse study. So this antibody blocks entry into cells, so you would expect it could prevent infection. But interestingly, it could also cure infection once it was already established, which was really unexpected. So this is in the context of the mouse model, of course. We have chronically infected mice. We give them the antibody. In monotherapy, it clears infection. So we think probably if you block infection of uninfected cells in the liver, then we have normal hepatocyte turnover, which eliminates the already infected cells and clears the infection that way. I think we've shown that our antibody can target the sort of genotypes of HCV that are more difficult to treat, genotype 3, for example. And we also know that it could probably block infection of the liver in the context of liver transplant. So we think, of course, depending on clinical trials, how that goes, that it could be a good therapy for these patients who currently don't have very promising therapeutic options. We don't really know. We know that HCV has a lot of diversity, and for some reason the current drugs don't impact genotype 3 as much. And it's also associated with more severe liver disease, like steatosis and faster progression to chronic long-term liver disease. I think not in the near future, but I'm hoping we can get there someday. The current therapies, as you probably know, are really expensive right now, so it definitely can't reach all patient groups. So we're working on alternative strategies. Other people are working on protective vaccines, which can also be really good. But I think we now have a good chance that we can achieve global control of HCV. I think when you're working with these new drugs in the lab, even in clinical trials, it's not really a real-world picture. In the real world, patients are much more complicated. They have other diseases ongoing. There's other socioeconomic situations involved. And so really, I think it remains to be seen how well the current drugs can actually address the real-world populations with HCV. Preclinically, it looks great. The next step is clinical studies, and we're very interested in proceeding forward with this, probably a phase 1 clinical trial to evaluate the safety to start with, and then we'll see how it goes. We're hoping to move forward with the phase 1 trial as soon as possible, but it's a bit complicated to find appropriate groups that we could test, so we'll see.